have with us Professor Anil Kumar Tyagi, Vice Chancellor Guru Gobind Singh Indraprasth University, New Delhi, as the chief guest of the symposium. May I request Professor N.K. Ganguly to kindly introduce our chief guest and invite him to deliver the inaugural address. Professor Ganguly, can we just give a small bouquet of flowers to our chief guest?
I think Professor Ganguly introduced me already in more details than I think I deserve.
Thank you, sir.
It's a privilege to have you with us, Professor Anil Kumar Tyagi. A round of applause for him, please.
एक मिनट सर for a photo.
Sure.
You will have to
I don't know which one.
Okay.
Don't do
Yeah,
it.
yeah,
It's
surely,
okay.
surely.
I'll just go ahead.
That I will do. Such a pleasure. Thank you.
It's okay. Everyone's watching.
Good afternoon. Professor Ganguly, Professor Nair, other members of organizing committee, dignitaries, guests, colleagues and friends. I am very thankful to Professor Ganguly and the organizing committee to have me here and give me this opportunity to present some of work that goes on in my lab. I must also confess that but for his invitation, I would not have read anything about probiotics. So that gave me also a chance to brush up something about probiotics. Thank you very much. I basically work in the area of tuberculosis for last more than 30 years. I mainly work in the area of vaccines and drug development. And today I'll touch upon little bit work on vaccinology related to TB. But before I get there, let me start with probiotics. Gut microbiota consists of a complex of microorganisms that live in our digestive tracts. The human body carries about 100 trillion microorganisms in its life intestines. And that's a number higher than the cells we are composed of. So it's a phenomenal number that we carry along with us. The four dominant phyla in the gut microbiota are Firmicutes, Bacteroidetes, Actinobacteria, and Proteobacteria. And if you ask where do they live, almost everywhere. In mouth, nasal activity, under the armpits, stomach, intestine, genitourinary tract, within joints, under the toes, between the toes, and everywhere perhaps. What kind of medical issues can probiotics treat? Probiotics may help in the treatment and prevention of a number of things. For example, infectious diarrhea, irritable bowel syndrome, antibiotics-associated diarrhea. They can help with the immune system. They can buckle it up. Eczema, diarrhea caused by Clostridium difficile bacteria, and ulcerative colitis. If you look at the mechanism of action of probiotics, the crosstalk between probiotic bacteria and the intestinal mucosa can do many things. For example, they can secrete bacteriocins or defensins. They can competitively inhibit other bacteria. They can inhibit bacterial adhesions or translocation. They can reduce luminal pH. They can increase mucin layer, and so on and so forth. In terms of immune system and its stimulation, the pathogen-associated molecular patterns derived from probiotic bacteria are recognized by pattern recognition receptors such as toll receptors or DCs in the epithelium, leading to downstream signaling and induction of Th1 cytokines. Of course, many parts of this still remain to be elucidated. Functions of probiotics are very large, and we can keep on counting for a long time. Stimulation of the immune response is one of them. They can be used as new vaccines. They can be used for treatment and prevention of acute diarrhea, restoration of non-intestinal microflora after antimicrobial therapy. They can be used for production of B vitamins, reduction of ammonia, cholesterol reduction, improvement of intestinal functioning, reduction of intestinal pH, positive influence in the intestinal microflora, increase of the lactose tolerance and digestion, vaccine adjuvant. I am not going to go through all the examples, but I will try to touch upon what I have mentioned in green, vaccine adjuvant and as new vaccines. But before I get there, let me come to the TB vaccines, and at the end of it, I'll show you how probiotics, at least in certain situations, can help even in the case of TB. Tuberculosis is a global health emergency. More than 30,000 lives are lost due to tuberculosis globally every week, putting TB in the list of top major killers. You can imagine a stadium full to the capacity with 30,000 people. That's how many people lose their life every week. 1/3 of the world population is infected with M. tuberculosis. The good news is that it is only asymptomatic in nature, and if the immune system remains robust, one can safely take the pathogen to its grave without ever getting the disease in active form. In India alone, more than 5,000 people develop TB every day, and more than 1,000 people die of the disease each day. 1/4 of the world's population, in terms of TB patients, reside in India, and that's a huge burden in our own country. TB, in terms of research goals, the most important goals for TB research or ultimate goal of TB research are development of new and more efficient and rapid methods of diagnosis, development of new vaccines for eradication of tuberculosis, and development of new anti-tubercular drugs which can reduce the period of chemotherapy as well as treat multi-drug resistant tuberculosis. TB is a complicated disease, and 5 decades of TB control program using potentially efficacious drugs have not reduced the burden of the disease in most part of the world. And a TB vaccine, which would be most effective in the controlling the disease, has eluded us all the time. Today, the only vaccine that is available against the disease is BCG, and BCG has produced more controversy than protection against TB. The largest trial against BCG that was carried out in Chengalpat area in South India proved that it has absolutely no efficacy against adult or older people. Although I must say that in all studies that have been carried so far, in children it has shown a great efficacy and prevention of tuberculosis. But as of today, we have no vaccine that can consistently provide protection in adults or older people. Thus, what we need is a vaccine which does what BCG fails to do, and fails to do for the reasons which we do not clearly understand. The new vaccine should protect consistently against adult tuberculosis. While this objective is unambiguously clear, the path to realize this objective has been very bumpy. And there are a large number of questions which are seeking answers. And some of them, although not all, are listed in my next slide. The nature of protective immunity in case of TB is not clearly known, although it is very important for a rational approach to develop a TB vaccine. Even to test the available candidate vaccines, as of today, the surrogate markers for vaccine-induced protection are not available. The role of animal models still remains an unanswered question because the protection obtained in any animal models or in all animal models does not guarantee protection in case of human beings. The host immune response is still an unanswered question because of the heterogeneity associated with it. Due to complicated nature of this disease, there is a lot of thought that perhaps in case of TB, we would require more than one kind of vaccine to control it. There is a dearth of lack of sensitive and specific diagnostic tools. And ultimately, after 90 years of its use, we still do not clearly understand why BCG fails in certain populations. That's where we remain. If we had a perfectly efficient or 100% efficient vaccine that would transform the TB control programs all over the world. But in the absence of answer to these questions, we do not have a perfect method to make a perfect vaccine. And that's why what is being followed today is a two-pronged approach which is based on the fact that on long-term basis, in order to develop a rational approach, you keep working for answering these questions. And in the meantime, based on the available knowledge and technology, continue to develop new TB vaccines, evaluate them in the existing animal models, and hope that some of them, based on the promise that they create, can be channeled for human clinical trials. This is the approach currently being used all over the world. There are several approaches that people have used, fundamentally following the principle that I mentioned to you, which are listed here. My own laboratory has taken approaches which are listed here in yellow: recombinant BCG approach, attenuated M tuberculosis strain, DNA vaccines, heterologous prime boost approach, and immunotherapeutic TB vaccines. I am not going to go through details of all the approaches that the laboratory works on. I'll take only one example. But this slide I like to go through because this summarizes all the work that we have done for the last 25 years. When we started this work, as Professor Ganguly mentioned, we started with the identification and characterization of mycobacterial promoters. Because at that point of time, it was not possible to express mycobacterial genes by using E. coli expression signals. So we worked on that for a few years. We then used in the second phase the knowledge generated from these promoters and the promoter themselves to develop expression vectors for expression of mycobacterial genes in mycobacteria. And one of the satisfying facts from this study was, apart from what we did later with these promoters and expression vectors, that large number of scientists in India as well as abroad have utilized these vectors for genetic manipulations of mycobacteria. Nevertheless, in the third stage, we developed recombinant BCG by using several antigens as listed here. At the same time, we also developed a number of DNA vaccines and evaluated both these series of candidate vaccines against subcutaneous challenge of M. tuberculosis. And I'm emphasizing that because at that point of time, the aerosol challenge model was not available in the country. But when we finished after 7-8 years of work, the first aerosol challenge machine in public sector became available in Jalma, Agra, and Dr. Katosh, being a very good friend, opened the door for us and said, you can come and use it, it's free for you. And for the next 6 years, we used that machine and the facility, and my students day and night worked there, and we re-evaluated most of the promising vaccines against by using aerosol challenge mode of infection. Finally, apart from that, we also developed some immunotherapeutic vaccines and attenuated M tuberculosis vaccines, and those were also evaluated. Some in Jalma, and later I developed a facility in South Campus, which is now working very well, and now we work at the South Campus itself. At the end of it, we developed 3 regimens. Fundamentally, if you look at, there are 2 ways of developing TB vaccines. One is that you can replace BCG. Second is you can boost BCG because it doesn't work in the older people. And based on these 2 approaches, as you can see here, we developed 3 regimens: recombinant BCG overexpressing antigen 85C, BCG expressing alpha-castelin gene, and boosted by DNA vaccines expressing the same antigen. And the third one on this side, boosting BCG, was enhancing the protective efficacy of BCG by boosting alpha-castelin based DNA vaccines. And these 3 regimens have been approved in principle by tuberculosis vaccine clinical trial expert group of DBT for eventual human clinical trials. And preclinical development studies for these are in progress. And what I'm trying to do is that only for one of them, which is this one, I'm going to show you some data before I end the talk and give you some idea how probiotics can be helped in terms of using in this context. Before I show you the data, let me just show you this slide on alpha-castelin from Mycobacterium tuberculosis. Alpha-castelin is a member of DOS-R, DOS-S dormancy regulon. It represents one of the most abundantly produced proteins during exposure to hypoxia, nutrient starvation, and transition of actively dividing cells to a dormant state. Latently infected individuals exhibit increased lymphoproliferative and interferon gamma response to alpha-castelin as compared to patients with active TB. These observations signify a crucial role of alpha-castelin in the elicitation of protective immune responses and maintenance of disease-free state in these subjects, thus making this antigen an attractive target for the development of new TB vaccines. At the top of it, you see the protocol for this vaccine. You immunize the animals with 5 × 10⁵ CFU of BCG. 6 weeks later, you boost the animals with 100 microgram of DNA expressing alpha-castelin. 6 weeks later, you challenge the animals with 50 to 100 CFU of M tuberculosis. And 10 weeks later, you subject them to euthanasia, as is shown here, 10 weeks time point. It's an early time point. And the vaccine efficacy is evaluated by bacillary load as well as by pathology. And as you can see here, that all the regimens that used here show effective protection as compared to the saline treated or unvaccinated animals, as you can see in this slide here. All of these you can see here. But the BBD regimens is the most effective one, and in compared to saline treatment, it shows 2.31 and 3.4 log reduction in the bacillary load compared to saline-treated animals. But more importantly, even in comparison to the BCG vaccination, it shows 1.37 and 1.96 log reduction in compared to BCG vaccination itself, indicating thereby that it provides a much superior protection than BCG itself. Apart from that, as you can see in the slides here, the BD regimen significantly reduced pulmonary, hepatic, and splenic lesions and provided enhanced protection in comparison to BCG vaccination. This shows pathology, and as you can see again, the saline group shows multifocal coalescing granulomas with extensive necrosis, or in the case of liver, multiple granulomas. In case of BCG group, it shows discrete granuloma with or without central necrosis. In case of BD group, there was negligible or diffuse aggregates of inflammatory cells, and there was no evidence of any inflammation in case of liver. We further extended this time period from 10 to 16 weeks to see the sustenance of this protection, that if we increase the time point, will the protection still last or will it still be lost? And the results are shown here. The protocol, by and large, the protocol is same, except that this time point of 10 weeks is made 16 weeks in this case. Now, as you can see here again, that in this case, the BCG does not function very well. It comes close to saline-treated animals. So when the time passes on and the time becomes longer, as is the case, BCG does not work very well after a long period of time. But if you look at the BD regimen, it still shows much better results, and it shows 2.1 and 1.47 log difference as compared to the BCG protection. So even in comparison to the BCG vaccination, the B by D regimen shows much superior protection even on extending the time period of euthanasia. If you look at the pathology or gross pathology of the animals, you find that the animals vaccinated with BD regimen showed a minimal involvement of lung and spleen and no evidence of gross lesions in liver in comparison to BCG immunized animals. As you can see here again, the saline group shows extensive multifocal coalescing granulomas with prominent central coagulant necrosis, as you can see in this case. And in case of BCG group, you have scattered areas of inflammation with discrete as well as coalescing granulomas with small necrotic centers. But in case of BD regimen, you have very negligible inflammation in lung and no evidence or any sign of inflammation in case of liver. We also measured vaccine-induced cytokines, both pro-inflammatory as anti-inflammatory in nature, to find out what kind of stimulation of cytokines occurs and whether they have any relevance in terms of prediction for some kind of markers. And what we found is that the degree of protection does not correlate with the level of any individual cytokines. It is more important to measure the relative emergence of cytokines in the cytokine milieu and consider the dynamic interplay of cytokines, which shows correlation with the protection. One of the parameters that always came to be true with the protection was that the degree of protection increases with an increased proportion of IL-12 and decreased proportion of IL-10. Now, due to paucity or non-availability of reagents in case of guinea pigs, we could not measure T-cell effector functions in case of guinea pigs. So we measured the protection again in case of mouse, and as shown here, as you can see here, in the mouse also, the BD regimen showed much superior protection than BCG itself, or in comparison to the saline-treated animals. And the vaccine-induced protection was associated with increased frequency of PPD and antigen-specific multifunctional CD4 T cells, along with higher production of interferon gamma, TNF-alpha, and IL-2. Now, I may mention here that when we finished these studies, there was an NIH team appointed by the Department of Biotechnology to review this work. And they suggested that while the work looks extremely good and interesting, before going to humans, we must consider the fact that although DNA vaccines work very well in case of animal models, they don't often work very well in case of human beings. So you may like to change that portion by keeping the antigen same. And that took us further in terms of developing an MVA construct expressing alpha-castelin as is shown here. It took us 1 year to make this construct because we were not trained in the virology. I had to get one of the students trained in the India Immunological in Hyderabad, and finally we made this construct as shown here, the expression of alpha-castelin. By using this construct, we then carried out the studies again. And as you can see here, the protocol was that you give BCG 5 into 10 to the power 5 doses. 12 weeks later, you challenge with M tuberculosis. 6 weeks later, you euthanize. In other case, you give BCG as such, but you boost it with recombinant MVA expressing alpha-castelin at 2 different doses, or with the wild type of MVA, or only saline-treated animals and do nothing because these are used as controls. The evaluation was carried about the same parameters again, bacteriological evaluation and pathological assessment. As you can see in this slide again, that all the regimens provided protection compared to the saline-treated animals, but the MVA, recombinant MVA expressing alpha-castelin, when used for boosting at the dose of 10 to the power 7, provided the best protection compared to all others, and it did much better than BCG itself, both in lung as well as in spleen. This is a gross pathology, as you can see here. The sham immunized animals exhibited maximal damage with numerous necrotic tubercles. Animals with BCG vaccination and those boosted with MVA-ACR exhibited minimal damage with very few small tubercles and normal size. This is the histopathology, and while sham immunized animals exhibited 70 and 20% granuloma in lung and liver respectively, their counterparts from both groups of vaccinated animals exhibited less than 20% granuloma in lungs and negligible damage in liver. Now, I want to mention that developing a candidate vaccine which works better than BCG in animal models has been extremely difficult. The reason for this is as follows. You want to develop a new vaccine because BCG does not work in human beings. But when we develop a new vaccine, you cannot test it directly into human beings. You have to first test it in the animal models. And in animal models, you have to compare it with the BCG. But in animal models, BCG works extremely well. That's why in animal models, to find a TB vaccine which works better than BCG is very hard. However, due to very hard work of TB investigators and due to large number of failures, there are more than a dozen of vaccines which are progressing in clinical trials, and I have shown most of them here, but new ones are regularly being added, and they belong to various categories like wholesale, subunit, live recombinant BCG, live attenuated cells, or viral vectors, and so on and so forth. It is true that based on the animal models, you cannot predict the worth of a TB vaccines. But at least on the basis of animal studies, I can say that the 3 regimens that we have developed look better than all the vaccines which are there in the clinical trials today. And although we can't say that we are beginning to see the end, but the things certainly don't look that bad at all. However, till we reach perfection, the efforts will have to go on. I'll stop this part here, and let me now come back to probiotics and show you an example where it has done beautifully well for TB. It is believed that tuberculosis develops because the body reacts to the invading bacteria with an overly intense inflammation which ends up being harmful to the body itself. The probiotic Niditum ricci, which is actually Mycobacterium manrescensis extract, is administered in pills as a food supplement that generates natural tolerance in the body against the TB infection. And TB is able to induce, it is able to induce a balanced immune response that diminishes the risk of acquiring TB, and the inflammation generated is much less. In a nutshell, the final dose consists of a heat-killed culture of an environmental mycobacteria belonging to Mycobacterium fortuitum complex, plus expecient. When administered daily for 14 days, the preclinical assay demonstrated that it is able to stop progression towards active TB. And I think this is a wonderful result. In another study, probiotics as vaccine adjuvants. Now, poor immunogenicity and limited ability to induce mucosal cell-mediated immunity are problems often associated with live attenuated, killed, inactivated, or subunit vaccines. Thus, enhancing mucosal immunity offers an effective strategy for preventing pathogen adhesion to host tissues. Pathogens such as Streptococcus pneumoniae, Corynebacterium diphtheriae, Bordetella pertussis, Haemophilus influenza, Enterotoxini E. coli, and rotavirus all enter host via respiratory or gastrointestinal mucosa, and therefore these infections might be subverted if mucosal immunity is enhanced. Probiotics have been shown to have pleiotropic effects on innate and adaptive immune responses, including modulation of disease, TNB cells, as well as antibiotics, antibody, and cytokine production. There are numerous evidences from human clinical trials wherein the use of probiotics has shown promising effects on vaccine-induced immunity. There are a number of examples listed here, as you can see, and I think this is something we are going to try with some of our vaccines. And I stop here. These are the people who have been working with me all over the time. And this group works on the drug part of what I do there, and this whole group works on the vaccine part. This Dr. Narayanan, Dr. Ramanathan, Dr. U.D. Gupta, and Professor V.H. Toj are my collaborators. And Vikram is the fellow who worked on Mycobacterium indicus pranae. He sequenced this organism. It was the first one complete sequence of a new bacterium from India. And I also acknowledge that we receive all the money for doing this work from the Department of Biotechnology. And it is all up because of these students that I'm able to present. I want to change this address when I give a seminar, and I still keep that same. Thank you very much.
Thank you, Dr. Tyagi, for a very illustrative presentation.